Hi, Micro Geniuses. I wanted to give you a video lecture on critical thinking and clinical applications for Lab 18. And it actually turns out that on Lab 18, there is no critical thinking and clinical applications. The lab is due tomorrow, Friday, the 20th of March. I want you to turn it in to me via email by 5 p.m. But I wanted to give you some hints. I am still working on your video lab for this week. It's going to be delayed, so you probably won't have to look at it until after spring break. I do apologize for that, but we'll keep rolling and I'll get on schedule just as soon as I can. But let's look at this. I'm going to switch over to the DocCam and we'll look directly at what you need to fill out and what you need to turn in. Okay. Here's the DocCam. There's my hairy wrist. And this is what we need to look at. Okay, so there are a few things that I need to point out here. First of all, make sure you put the purpose of the lab and write your unknown number. You had unknown 1 or unknown 2, so U1 or U2. And then this next table here, skip. Don't write anything. We didn't do that. We didn't do that at all. So what you need to do is you need to make a table listing the test you performed and the results. Oh my goodness, I don't know if you can necessarily do this without your laboratory notebooks, which you turned in. I just thought about that. But if you could possibly do your best to remember those tests, and if you don't remember, then go ahead and indicate with a question mark. And we'll just do our best. Okay, then if you look at the next page, okay, then that says, what is the organism shown in figure 18.1? Okay, and that's an interesting one. So figure 18.1, go back here, and you have all sorts of different tests for 18.1. You know that it is an unknown organism, and from a culture perspective, on a culture plate is yellow. On a fermentation tube, it does not ferment glucose, okay? It doesn't use glucose. Otherwise, that fermentation tube would be yellow. And then next we have a gram stain. And although this is really difficult to see on a webcam, it's purple. Then you have a lipid hydrolysis test showing that the bacteria can hydrolyze lipid. These are, this is not a Kirby-Bauer test, but these are colonies of bacteria, and you see clearing where lipid was in the media, sort of like the starch hydrolysis test, only instead of looking at starch, you're looking at fat. So this can hydrolyze lipid. And then finally, we have the catalase test, and you can see that it bubbled and it's catalase positive. So what we do to identify this particular organism is we go back to Appendix H, which I believe you've used before, and you look at keys to bacteria. Okay, so we go here, and if it's gram negative, then you use this particular table. If it's gram positive, whoops, then you use this particular table, and so that will help you to identify the organism, okay? And you want to be able to identify that. Then, going back to the lab 18, is then you identify what was your unknown, okay? So make sure that you put your number and then the actual genus species of your unknown. And then on a separate sheet of paper, actually down below, you can do this here. Then you want to write the rationale for your conclusions. And then you want to write why is it necessary to look at the physiology based on this particular lab rather than just morphology, and why that's necessary to distinguish between the variety of bacteria that we've been studying. So I hope that helps. I just had an idea, and that is that I'm going to try to scan your pages in your laboratory notebooks today before I leave, and get those to you. If I can get those to you today, then the lab is due tomorrow. If I don't get those to you until tomorrow, then I'll just make it due after spring break. But you know, there is a way to get these to you. I just have to scan all 10 laboratory notebooks, the page that you had for lab 18. So I'll go ahead and do that. I've got your lab notebooks. Don't worry if I don't get it done today. Don't worry about turning it in tomorrow. And I hope you stay safe. And if you have any questions, you can shoot me an email, or you can Skype me at brhooker in Skype during my office hours, or call my desk phone at 530-226-4734. Thanks a bunch.